Hey all, today we're going to talk about genomes and some biotech. So, what is a genome? A genome is the complete set of genes or genetic material that's present in a cell or organism. So, this can include the genes, which are the coding regions, and also non coding regions. You can also have genomes for mitochondria and chloroplasts. The Human Genome Project was completed in 2003, and it was a research project whose mission was to decipher the chemical sequence of the complete human genetic material, so the whole genome. So they wanted to identify all 50,000 to 100,000 genes contained within the genome, and that provides some tools to be able to analyze that genetic information. Eukaryotic genomes in general are pretty large, much larger than prokaryotic genomes, and they have more protein coding genes. And this is largely because eukaryotes generally have many cell types that are going to require specialized proteins. Eukaryotic genomes also have more regulatory sequences and therefore the production of regulatory proteins because they have much greater complexity requiring greater regulation. So they have many more points of control in the expression of genes. Again, this is largely due to the higher likelihood for cell specialization. And also, eukaryotic genomes have non-coding sequences or DNA sequences that are not going to be transcribed into functional RNAs and therefore translated into proteins. Some of these non-coding regions are repetitive sequences. So these are highly repetitive sequences that do not code for proteins. So these are short sequences, less than 100 base pairs, that are repeated thousands of times in tandem, so right side by side, but are not actually transcribed. The proportion of these repetitive sequences varies in eukaryotic genomes, 10% in humans and then 50% in some fruit flies. Short tandem repeats are something that we're going to talk about with biotech analysis. Short tandem repeats, or STRs, are sequences that are 1 to 5 base pairs in sequence and then repeated. And they can be repeated up to 100 times at an individual chromosome location. So various STRs at particular locations are going to vary in length. So you can have the same STR at the same part of your chromosome, but that STR is going to vary in length from you to another person. And then you have a whole bunch of those different STRs. Moderately repetitive sequences are typically repeated about 10 to 1,000 times in the eukaryotic genome, and these include sequences that get transcribed to produce tRNAs and rRNAs, so those helpers in translation. This diagram right here, this is looking at gene functions in Drosophila, or fruit flies, and these are pretty typical of many complex organisms in terms of the percentage of different genes. So let's get into some biotech analysis in terms of crime scene analysis. So when you're analyzing samples from a crime scene, you're often looking at really small genetic samples. So you might only be able to isolate a very small amount of DNA. And something that you're often faced with when looking at a crime scene is possible contamination. So if some of your sample has been contaminated, you might be working with a really small sample. When we're doing a crime scene analysis, what we're actually really looking at is doing an STR analysis. So if you remember, an STR is a short tandem repeat, and it's 1 to 5 base pairs and can be repeated up to 100 times at a chromosome location. So one short tandem repeat, an STR, can have a different length than another STR, depending on how many times it's been repeated. So here is a look at different STRs on chromosomes, right? These are the 13 that mostly get looked at. So here's an STR on chromosome 2, here's another STR on chromosome 3, and so on and so on. So what STR analysis does is it wants to first isolate those short tandem repeats. So kind of cut those short tandem repeats out of those chromosomes and just look at those repeating segments. And this is looking at measuring the exact number of repeating units. So you want to essentially measure the length of that STR based on how many times it repeats. And one individual STR, so like this one per se, could repeat 10 times in one individual, and it could repeat 20 times in another individual. So there's going to be some variability in STR lengths from individual to individual. Now, for forensic analysis, you really look at these 13 STR loci in this what we call combined DNA index system, or CODIS database. So there's going to be variability in the STR lengths at each individual place for TPOXX versus this one, FGA. But then if you think about looking at all 13 at once, unrelated people are almost certainly going to have different numbers of repeat units. So it would be extremely unlikely that two individuals would have the same length STRs for all 13. You can isolate these STR segments using specific primers that will actually cut them out, and we'll look at that in a second. And then you amplify those segments, or you make more copies using a technique called PCR. And then you would visualize the STR segments using gel electrophoresis, which we'll talk about in a second. This video by Bozeman on STR analysis is posted on the portal and is definitely worth a watch. So how do you cut out the STR fragments? Well, first you use a restriction enzyme, and the restriction enzyme is this right here, EcoRI, which is a restriction enzyme found in E. coli. And what EcoRI does is it's looking for a particular sequence. So in this case, it's looking for this sequence here, AATT, and then TTAA on the other part. And it's actually going to cut the double-stranded DNA molecules into smaller fragments by breaking the bonds of the DNA backbone between the 3' hydroxyl group and the 5' phosphate group of the next nucleotide. So breaking this bond right here, breaking this bond right here. And it's going to cleave the DNA, it's going to break the DNA at those specific sequences of bases called recognition sequences or restriction sites. So in this case, the restriction site is AATT and then TTAA. That's the restriction site. So that's where the restriction enzyme is targeting. Most of these restriction sites or recognition sequences are going to be about 4 to 6 base pairs long. What happens then is the way that the restriction enzyme cuts it leaves it with these sticky ends. So like here's a sticky end, and then here's a sticky end right here. So it's going to cut the DNA strands so that there's going to be a short sequence of single-stranded DNA at each cut end. So you kind of have this like single-stranded overhang. And then that single-stranded overhang is able to form hydrogen bonds with then some complementary sequences on other DNA molecules. So in this case, you're going to be replicating both of these sides. You're going to replicate this side and you're going to replicate this side, but this sticky end right here is giving somewhere for that replication to start. So this replication would move that way largely, and then this sticky end right here is going to give a spot for replication to start, so this replication is going to move in that direction. So this process can also be how you can get recombinant DNA. So you can kind of like chop out DNA sequences and then insert them into other DNA sequences. Most of the time you're going to use this sticky end way of doing things, but you could also do like this where you have blunt ends formed, which is going to be just a little bit different in terms of how it's being replicated. You make cuts that are directly opposite one another on the two DNA strands instead of having those single-stranded parts. It really largely depends on what enzyme, what restriction enzyme you're using. PCR, or polymerase chain reaction, is a technique that's used to amplify DNA segments. So essentially just make a lot more copies of the DNA segments. PCR is accomplished by thermal cycling. So what you do is you heat the DNA up. So first there's the denaturation step. So this is where you heat it to about 95 degrees Celsius, right? And what that does is it separates the two strands of the DNA. So now you have the two strands separated, and then your next step is annealing. So this step, the annealing step, is where you have primers attached to the DNA strands, so the two separated strands. You have primers attached, and those primers are specifically targeted for the DNA sequence of interest. So if you're looking at one SDR region, those primers are going to be targeting just that region to just copy that part of the DNA. And that's done at a cooler temperature, so 55 to 65 degrees Celsius. So first you heat it up to denature it, and then you cool it down so that the primers can attach. And then the next step is extension. And extension is done at 72 degrees Celsius, so another temperature. And in an extension, DNA polymerase is going to attach to those primers and then just copy the DNA strands along. And I think it would actually be really, really helpful if you guys can watch this polymerase chain reaction video on the portal. It's on the topics page, because that's going to give you a much better idea of how the polymerase chain reaction steps are going to work to isolate a very specific strand. Because it's actually going to take a few rounds of PCR in order for you to just isolate that one section that you wanted. So it's going to take several rounds until you have a greater proportion of the DNA produced be just that targeted region. So watch that video so you can get a better sense of the process, and then we're going to try to do some paper PCR in class. Once you have all of your STR fragments or your DNA fragments kind of cut out and then amplified, so made a whole bunch of copies of them, you're going to run those fragments through gel electrophoresis. And what gel electrophoresis is going to do is it's going to separate out those DNA fragments by size. So you can determine the number of fragments that you have and then the size of the fragments by base pairs, number of base pairs for each fragment. So those DNA samples will have had to be dyed with a fluorescent dye, and then the samples containing the fragments that are dyed are going to be placed in wells at one end of a semi-solid gel, right? So the wells are going to be down here, right? So you're going to place those samples in the wells. That semi-solid gel or agarose is a porous gel. So then you're going to take that porous gel, you're going to put it under a buffer. All of your samples are in the wells. So it kind of ends, if you're looking at it sideways, it looks like this. It's kind of lying in this compartment, right? And then it's covered by buffer solution, and then your wells are going to be at this side, and then your samples are in the wells. If you're looking at it from above, right, here's the gel, and then you have all of your wells at one end of the gel. You're then going to apply an electrical field through the gel. So what you're going to do is you're going to place the positive pole down here, and you're going to place the negative pole up by where the wells are. And because of the phosphate groups, DNA is actually negatively charged at neutral pH. So if DNA is negatively charged, it's going to move, the DNA fragments are going to move through the gel towards the positive end of the field. And what's interesting about this is the smaller DNA fragments are able to move faster than the larger ones because the spaces in the polymers of the gel are small. So in the gel is porous again, so those spaces are kind of small, so the smaller DNA fragments are going to be able to move faster. So what's going to end up happening is your large fragments are going to end up closer to where your wells are, where that negative pole is, and then your smaller fragments are going to end up down here where the positive pole is on the opposite side of the wells. You can then compare the size of the fragments that have separated out to a DNA ladder, which is something where it's chunks of DNA that are cut up that are known, so it's known DNA sizes. And you visualize those DNA fragments using a UV light. So what you're looking for is you're looking for the number of fragments, right? So, and you can look at the number of fragments produced with the restriction enzyme. It's going to depend on how many times that recognition sequence occurs in that particular sample. And then you're also looking at the sizes of fragments, right? And you can then compare the DNA fragments to the DNA fragments of known size or the DNA ladder to look at really particular size. And then you can also look at the relative abundance of a fragment or how much DNA is present, and that's going to be looking at the relative intensity of a band produced by a specific fragment, so how dark it is. What we're really going to be paying attention to in our crime scene analysis is the number of fragments and then the sizes of those fragments. So when we do this in the lab, what we're going to have is we're going to have our DNA from our crime scene, which is this right here, and then we're going to have DNA from 3 different suspects. So once all of our DNA separates out, we're going to then compare the DNA suspects to the DNA from the crime scene. And what we would expect is we would expect our suspect of interests, we would expect their DNA to be the same number of fragments and same size of fragments as the crime scene DNA. So I'll give you guys a second, pause the video if you need to, and see if you can determine which of these DNA samples, so which suspect in this case, matches our crime scene. So if you guessed suspect number 2, you were correct, because their DNA bands closely match the crime scene's DNA bands. Some other places where restriction enzymes and recombinant DNA are kind of interesting. One is CRISPR, so this is with gene editing. So what scientists are doing with CRISPR is they actually design and synthesize these short RNA molecules that are going to match a specific DNA sequence of interest. So this is called guide RNA right here. And it's looking at it's trying to match up to a particular DNA sequence. So the guide RNA shuttles this molecular machinery to the DNA target, and then when the DNA target is found, you have this enzyme, Cas9, and that actually will bind to the DNA at that part and cut it, and then it could potentially shut that targeted DNA off. Modified versions of the Cas9 enzyme can activate a gene rather than cut it. CRISPR-Cas9 can modify like typos, so it can like treat genetic diseases in that way. It could silence a gene or actually change the sequence of a gene once it's able to get in there. Somewhat similarly, GMOs, or genetically modified organisms, these are transgenic, where you're actually combining genes from 2 organisms, so you have altered genomes. And this is, again, using recombinant DNA technology. So identifying a DNA target sequence and then cutting that sequence using enzymes, and then you can actually swap in a gene, or you could just take that gene out. What's interesting about GMO foods is there's actually no evidence that they're less safe than non-GMO foods. There's no evidence that they cause cancer or celiacs or autism or diabetes or obesity. They can lead to more sustainable agriculture, like higher crop yield, reduced need for herbicides and pesticides, improved food quality. And a lot of this reason is because, say you have one crop that just has a natural resistance to pesticides, and you have another crop that is not very resistant to pests. What you do is you take that gene from the crop that is resistant to pests and you insert that into the crop that has low resistance for pests. In some cases too, you can actually engineer foods for higher nutrient content. So in this case here, this rice has been genetically engineered so that it produces more vitamin A. They call it golden rice. So it's able to biosynthesize this beta-carotene gene, and that gene is coming from daffodils and bacteria. There is some allergy worry in terms of GMOs. Potentially inserting a gene from an allergen could potentially cause a new food to express that allergy if you've inserted that inserted that gene into the new food. There's also some worry that it could have effects on the surrounding environment, like crossing with wild organisms, potentially creating superweeds, maybe having problems with food chain effects, or even maybe affecting bee populations. But there are some really interesting examples of GMOs. One is they've got goats producing this human protein in milk to treat a blood clotting disorder. They've also got a vaccine producing bananas, so you could maybe have edible vaccines. They're producing blue roses by taking the gene from pansies and inserting it into the roses genetic code. And also glowfish. So here's some glowfish, and these are zebrafish that have had the fluorescent protein gene from jellyfish inserted into their genome so that they're actually able to glow. Another option is actually doing gene therapy. So you have a couple options here. One is gene augmentation therapy, so right here. And this is where you might have a cell with a non-functioning gene due to a mutation that causes some disease. And you can actually add DNA from the functioning version. So you can kind of add the functioning gene back in to the cell. But these are only successful if the effects of the gene are going to be of the disease are reversible. So this is often more successful in disorders like cystic fibrosis, which are loss of function disorders. You can also have gene inhibition therapy. This is looking at treatment of infectious diseases where you're introducing a gene that inhibits the expression of another gene. So you kind of introduce a blocking gene, you can call it, right? So here's your blocking gene. And that blocking gene is going to inhibit the expression of some other gene, or it will actually interfere with the activity of a product that gets produced. With the case of cancer, it could potentially eliminate the activity of a specific oncogene. That's an option. Another option here is actually trying to kill specific cells. So you can insert a DNA into a diseased cell that causes that cell to die. They call this like the suicide gene, where that gene is going to produce some toxic byproduct, or the DNA that is inserted causes expression of a protein that causes the cell to be marked by attack for the immune system. In some cases, the transferring of these DNA sections, they can actually get packaged within a vector. So the vector could be a virus, it could be a bacterium, it could be a plasmid, so those little small rings of DNA. And that vector is going to carry that DNA to the cells, and then once inside the cells, that DNA or gene will be expressed. Some of the challenges for this, you want to make sure you're delivering the gene to the right place and turning it on. You also want to avoid immune responses. Some of these vectors might be more or less likely to trigger an immune response. Another challenge right now is all of this gene therapy is really expensive. So that's it for now. So I would check out those 2 videos that are on the portal, and I will see y'all in class.